So good afternoon, everybody, and welcome back from the lunch break and the AGM. My name is Clive Page, and I have the pleasure of chairing this year's Paton Memorial Lecture, my colleague and friend, Professor Sue Brain. I'm sure many of you know Sue and her work, but just for those of you, perhaps younger members in the audience who don't know Sue, she's a professor of pharmacology at King's in London. And Sue was also my head of department, I think. I can't remember how many years, Sue, but a lot. She was the head of the vascular biology and inflammation section in our cardiovascular medicine and sciences group. She's the lead for development, diversity and inclusion for our school at King's. She's an honorary fellow of the British Pharmacological Society, and she's held a number of posts within the society. She was Vice President Academic Affairs. She chaired the BPS Education and Training Committee, and if you were at the AGM, you'll have learnt she's just stood down after 6 years of service for the Society on our Finance Committee. Sue's also served on a number of grant committees, and she was a member of the REF 2021 committee, which is something that I know took up a lot of your time, Sue. And I think above that, Sue's trained lots of postgraduate students over her career. She's a chartered science teacher, and she's a formidable teacher who's taught students at all levels. This afternoon, Sue's going to talk about one of her major research interests, which is the role of sensory nerves in vascular inflammation, including the role of trip channels and many other pathways, and particularly CGRP and vasoactive peptides. And it gives me great pleasure, Sue, to introduce you as this year's Paton Memorial Lecturer. And Sue's going to talk to us about sensory nerves and their neuropeptides from discovery to new medicines. And just to remind you, there will be questions at the end. Sue's happy to answer questions. But if you don't want to stand up and ask them publicly, please do put them on the app, and we'll capture them here and deal with it afterwards. So, Sue.
Okay, well, thank you very much, and it's a great honour to be able to give this lecture. So, I'm going to talk about sensory nerves, commonly known as the pain fibres, mainly C fibres, A delta fibres, etc. So, their history is complex, and the discoveries are not linear. And at times, several people have appeared to be working on similar things, and I've realised that they haven't really discussed the subjects too well across theologies, so I've learnt a lot. Some discoveries and scientists are well known, and others have remained in the background. So what I want to talk today is to go over the history of sensory nerve pharmacology, really, of capsaicin and related molecules, discuss the sensory neuropeptides, some of the major ones, and look at where we are today. So, sensory nerves have been known about for a long time, indeed about 300 BC, when this was meant to have taken place in Turkey, I believe. Herophilus of Chalcedon realized that motor nerves were joined to muscles and sensory nerves to organs of sensation. This is a painting from 1906 where he's on the right there teaching anatomy. That was his main interest. More recently, we've learnt about how the sensory nerves function. In 1874, Goltz realized that sensory nerves actually, if you stimulated them, would cause vasodilatation. Stricker realized if you stimulated the sensory nerves, they would contribute to the inflammation and the vasodilatation. Then Sheraton, a physiologist, realized that there was a sensory spinal skin field. He studied skin. This is relevant to a lot of other tissues. And Bayliss supported and extended about this, talking about antidromic vasodilatation, where stimulation in a tissue-like skin will lead to an axon reflex flare. And Langley talked about this, calling it antidromic action. Another notable figure, not a pharmacologist, was Sir Henry Head. And he, with colleagues, put together a knowledge based on the pathology of herpes zoster and its bearing on sensory localization. And these led to the dermatome-type figures that are used today for people to understand, if you take an area of skin, where it's linked up to in the spinal cord. He identified that sensory nerves really play a pivotal role in inflammation. It's classic work, but the physiologists don't seem to mention it, or the pharmacologists too much. But I think these studies are unique, as they involved people who died at institutions where they'd previously had herpes zoster. So he compared lesions and rashes from clinical notes with those of the nerves and ganglions that they innervated post-mortem. It is impossible to do today. People still depend on his work clinically, and it was an amazing thing he did. Even more amazing, this picture shows him and his arm next to a model of an arm and a colleague. And he liked to experiment on himself. So he actually took his sensory nerve in the arm, got his colleague to ligate it, and then he went on to work out how long it took to regenerate. And during this time, he went to Germany to get special tools so that he could measure the sensitivity of the skin. They were the only place he could get them in the world. I always, and I've told a lot of students this, I've always thought Sir Thomas Lewis, who was based at UCL, was the first clinical pharmacologist. So I've learned through preparing for this lecture I was wrong. He was basically a cardiologist, and he developed the echocardiogram. So he was a great person before he started in later life to work on responses of the skin to injury. He looked at histamine especially, and histamine-like substances, and developed the concepts of the triple response. So what I've got here is saline injected skin vehicle, and then here, hopefully you can see there's a wheel and also an axon reflex flare. So there's a triple response, and this was taken shortly after a histamine-like agent was injected intradermally. So you've got local reddening, edema formation due to the wheel, and the axon reflex flare. And this he developed the concept of the triple response with, and it's the basis of allergy testing still today, although admittedly a lot of people now use prick tests. So moving on, we understood sort of quite early on that sensory nerves were important for, of course, taking pain sensation to the spinal cord, but also having other activities, including the axon reflex flare and vasodilatation and edema formation. But I'm going to sort of go a little bit sideways now and talk about a pharmacological tool which has been so valuable to this area, and that's the chili extract capsaicin. It's been pivotal for exploring physiological functions in sensory nerves. So it's been around for a long time, and in 1816, Bolshoff isolated it. Thresh then isolated the crystals and named it capsaicin. Space used a capsaicin-like substance to show that it was related to the sensory nerves. Then Bruce realized that you needed an intact nerve supply from his studies in the eye. Then Breslau studied these reflexes in skin, and more recently, a growing number of people have studied them. In the 1950s to 70s, the Jasko family realized that capsaicin caused a burning pain, and Sholzani started to work at that time as well. It has been, as a consequence from this burning pain, utilized for defense in pepper sprays. It has a short duration and few adverse effects, but if you've got capsaicin in your eye, you'd call it quite a major adverse effect, and that's one of the problems with capsaicin. So Sholzani has played a major role in studying sensory efferent function, and realized that you had desensitization on continued application, that there was an ultra-potent agonist to capsaicin, and with these desensitized effects, you could get potent potential analgesics. There's been a lot of others who've studied in this area, Salazzi, Bevan, and others, and they realized capsaicin must have a binding site, and they developed the first antagonist based on this. Others studied the gut sensory roles, and others eventually ended up studying the cranial circulation. So there's been capsaicin creams that have been utilized therapeutically for a long time. There's also been these creams used illicitly, for example, in equestrian circles to deaden horses' legs so that they jump higher or run faster. And when a capsaicin test was developed for the 2008 Olympics, 4 teams that reached the show jumping finals were removed. So, without a doubt, capsaicin appears to work as a painkiller when it's used in a desensitizing regime. I thought this probably came from the 1950s, but a couple of years ago, I was lucky enough to carry out a sabbatical at the Department of Pharmacology at the University of Otago with Debbie Hay. And in their seminar room, they had loads of bottles. So I loved looking at these old bottles, and I found one which was Evans witch oil. And that contained something which was 33% capsaicin with a calming agent, just a little bit of a calming agent. In 1908, this particular witch's oil won a prize in Dunedin. So it won a gold medal. And what I found absolutely amazing about this was that at that time, they realized it stimulated nerves and vessels of the skin. They thought it helped the blood circulation, and that's why, as they put it, it could cure all ills. And they said it was really important for heads of families to have a bottle of this in their family home. It looked quite expensive to me for the time, 4 shillings and 6pence. But what it made me realize was that people have known for an awful long time that capsaicin can be used as an analgesic. More recently, coming up to about 2010, capsaicin patches, which are 8% capsaicin, have been developed, Cutenza. And the idea is that they are an effective treatment of neuropathic pain for quite a long time after application. There's been a meta-analysis of Cutenza treatments, and it's highly effective in more than one type of pain, of chronic neuropathic type pain. But it's never been as popular as I expected it to be. I thought this would be a great new painkiller when it came around. And I think through reading a systemic review, which has come out this year, I understand why. The point of the review was to see if it was as effective in old as young people. And it was, that's good. But the application site suffers a lot of discomfort. So 36% in this systematic review had beneficial responses with neuropathic pain syndromes. And that's, based on my experience, good if a third have beneficial effects to a medicine. But 81% have adverse effects. I assumed it was where the person putting on the patch would get it in their eye or something like that, but it seems to be that these patches can cause blisters, etc., and skin discomfort. About 80% of them are serious. It doesn't seem to lead to that many people discontinuing treatment, but I suspect it stops a lot of clinicians from suggesting the treatment in the first place. But we've reached a point now where there certainly are more drug discovery programs in progress for the use of capsaicin and ultra-potent agonists. Okay, they may be applied topically, as here, or they may be injected. And some people have suggested when they're injected, say into a knee for OA pain, that they may not have the instant burning pain, but they may cause a long desensitization. So that's an interesting area, I think. Of course, as I said, the capsaicin binding site caused a lot of interest, and it wasn't long before the TRPV1 receptor channel was discovered by two groups simultaneously, one in the UK and one in the States. After that, there was a huge amount of work, and I've put some of my papers here where we studied it in mice, in arthritis and pruritus, and in terms of maintaining body temperature. This naturally led to a drug discovery program, and a lot of companies had TRPV1 antagonists available for testing. But they've never been very effective. Okay, they're analgesics, but they do have adverse effects. It may be in regulation of body temperature, they're hypothermic, but this has been got around in some cases, or in terms of noxious heat sensation. So when you're taking something out of the oven, you can easily burn yourself because you just don't feel the heat. So that, when I did a survey last year, I couldn't find any that were still going through the drug discovery process. But suddenly, a year on, we have topical TRPV1 antagonists, rather like the Q-tensor patches, that are reaching clinical trials for a range of conditions, and including post-operative pain and pruritus. And these agents, from what I can see of what's written, seem to be well tolerated and beneficial. So we could have a whole range of patches coming up that should be effective as analgesics that hopefully won't have the initial burning pain, because these are in the main competitive antagonists, and they don't activate initially. So we've done an awful lot of pharmacology in my mind with respect to capsaicin and related compounds. But what about the actual sensory nerve itself? Are there substances that it releases? And the answer is most definitely yes. And I'm going to talk about substance P first, because it was the first neuropeptide, 11 amino acid peptide, member of the tachykinin family. And in 1931, von Euler and Gadam realized that there was something that wasn't blocked by atropine. They didn't have many blockers at the time, but it certainly wasn't sort of parasympathetic-like. I think von Euler was quite lucky. He seemed to be on a visiting fellowship for a short time from Germany, and he discovered it. It was soon called substance P. The P is for powder, and most of the studies at the time were done in equine brain and gut. And Lembeck, later on, in Austria, realized that sensory nerves mediate neurogenic inflammation. And then in 1971, Susan Lehman isolated it and realized the 11 amino acids. I've got a picture of Susan here, because when I was doing this research, I found something that was quite amazing, in that of all the historical people, she's the only female. I really am shocked about that, but somebody said to me, well, that's the way it was then. But not now, thank goodness. There are other people who had major contributions. Hochfeld, who through making antibodies, carried out immunohistochemistry and realized substance P was released from sensory nerves. Lembeck and Holzer, they realized that substance P mediated the neurogenic inflammation that's from sensory nerves. And it was later realized that capsaicin treatment degenerated, and then you wouldn't get the neuropeptides released. So everything fitted together. And then some of the first peptide antagonists came along, and you could inhibit this response. This was exciting for people because there were thoughts that neurogenic inflammation, which is relatively acute, actually could lead to interactions with the immune system, and as well as pain, and there was another drug discovery pathway. This is where things go a little bit wrong. So there's substance P receptors, there's NK1, NK2, and NK3. And they were studied all for either inflammation or for perhaps some sort of CNS effect, first of all. The first ones that came for clinical trials were NK1 receptor antagonists, very selective agents that worked really well in preclinical trials and animal models, but they failed in clinical trials related to pain and inflammation. This was migraine, arthritis, peripheral neuropathy. It was thought they may be antidepressants, but that eventually failed as well. I've talked to several people who've been involved in drug discovery programs for these antagonists over many years. And they really couldn't believe the results. Indeed, I think there's still people in the BPS who are investigating why they failed today. A couple of people are Leslie Iverson and Stuart Bevan. But there was a role clinically for an NK1 antagonist. Apretapunt is probably the best known, and it's antiemetic. This was eventually realized through preclinical pharmacology. There's been a systematic review just recently to show that apretapunt outperforms ondansetron. I must admit, I thought apretapunt was given when ondansetron didn't actually work to control nausea after cancer treatments, but perhaps there's more of a role for the NK1 antagonist than I thought. The NK2 and NK3 receptor antagonists that were developed, it was thought would be beneficial somehow in the CNS, but that didn't turn out to be so. But just a couple of weeks ago in Nature, there's a study of NK2 long-acting receptor antagonists, and it's in rats and macaques, and it shows that NK2 receptors not only decrease body weight, but seem to reduce diet and seem to help with metabolic resistance. All the things you might associate with GLP. So they need to study the mechanisms further, but this is quite an exciting thing going on, and I don't think the studies should, I'm so pleased the studies have gone on for as long as they can with these receptors, because there's an NK3 receptor antagonist that has now been cleared, certainly by the FDA, for clinical trials for menopausal flushes, and is now marketed for these flushes. There's a CNS action there that's very selective with the NK3 receptor, and there are very few other drugs for those people who suffer with menopausal flushes. So there's huge excitement there as well. So substance P went from being something that was very disappointing to I think it's now a lot more exciting in terms of drug discovery. What about other neuropeptides? You can find about 50 in total with various splice variants and other things. There's an anti-inflammatory one called somatostatin, studied by some of our Hungarian colleagues who are at the meeting. But there's also calcitonin gene-related peptide, or CGRP for short. And that's one I've worked on quite a lot. And it was discovered in the early 1980s. It's a 37 amino acid peptide. There's an alpha form, which is the major vascular phenotype. And it was one of the first peptides to be discovered through molecular biological techniques. Of course, it's contained in and released from the same nerves as substance P, although there's some other nerves where it's released alone, potentially some of the A-delta fibers. This is the first study that I was involved in when we realized it was a vasodilator. I worked with Tim Williams, and this is his arm, and he was determined to see if it was active in a human. So I injected a dose intradermally. This was all ethically approved at the time, you can see a darkening there due to increased blood flow. What we think's happened is that it's got into the lymphatics, and we've got these little legs, looks a bit like a scorpion or something like that, where the CGRP in the lymphatics has innervated around the blood vessels. This picture was taken 60 minutes afterwards, and it's a very different response to the response to histamine, the triple response that you'd expect, and no response to vehicle. Down here, we've got something that's more pharmacological. We've got decrease in doses of CGRP, and I hope you can see with this dose that there's erythema there due to increased blood flow, and that's the amount you'd expect to be, or the amount you'd expect to be released in the human skin. Substance P, very different. You get the triple response just like histamine, and then it causes edema formation in the human skin. No local reddening or anything like that. So, what have we done to try and understand more about what these peptides may do together? Well, we've used an assay called the mouse skin assay that Dipesh Thapa was talking about yesterday, because we've since developed it into a class practical for undergraduates. We use this assay because Tim Williams had developed a two-mediator hypothesis, and we wanted to see if substance P and CGRP fitted this two-mediator hypothesis. Basically, it involves anesthetizing whatever species, this is mouse, injecting Evans blue IV, which binds to albumin, then shaving the dorsal skin and marking sites out for injection, then watching as dematous sites, they'll turn blue due to the albumin bound to Evans blue. When you do this, you find that vehicle, Tyro solution was used here, does very little. In vasodilator doses, CGRP does very little. Substance P, as I said, is a mediator of increased microvascular permeability and will cause some edema formation. But it's not a vasodilator, and to get the true acute neurogenic inflammation, you need both substance P and CGRP. So you get arteriolar vasodilatation in skin due to CGRP, and then in the post-capillary venules, if they're leaky, you'll get a potentiation of edema formation and also cell accumulation as well. Tim Williams, FRS, made his name through the study of prostaglandins at the time and mediators of increased microvascular permeability such as bradykinin, histamine, complement factors, and things like that. It worked really well when I used this for substance P and CGRP, and we've since adapted it for use in mice and also adapted it for the sorts of agents you get available to you today. We went through a phase of using radioactivity. We can't get that anymore. So this assay has been a fantastic assay for us to understand a little bit more about neurogenic inflammation. It took a long time to understand the CGRP receptor. There were some false starts, and I think there's some papers that ended up being having to be removed from the literature. But eventually, it was realized by Steve Ford and colleagues at Glaxo that it was a G protein, and it had to have with it, for a CGRP receptor, a receptor-associated modifying protein. For different related peptides, there's different receptor-associated modifying proteins. But the peptide actually sits between both the modifying protein and the G protein. There's a receptor component protein which may aid signaling, but this is being debated now. And then, most normally in the tissues I work with, you get an increase in cyclic AMP and vasodilatation. So, it wasn't long before we knew quite a lot about CGRP, that it could act locally in skin and other tissues. And it could also act systemically, perhaps because it's a hypertensive agent or perhaps because it may be protective. Very soon after we realized its vasodilator activity, people started looking at the cerebral circulation and specifically thinking about what it may do there. And Gosby and Edvison had already done an awful lot of work on migraine by this time, And they were involved in the study of substance P and realized that substance P antagonists didn't benefit migraine, so they asked the question, could CGRP be the substance that they did? So, in trigeminal sensory nerves, you have release of CGRP and vasodilatation, and I've got a blow up here. And here, the CGRP is being released from nerves to act on smooth muscle cells. It could be a nerve here that they act on as well. So, if this is important in migraine, what could we learn about this scenario? Triptans were the first specific treatment for migraine, and triptans were found to inhibit release of CGRP. And then it was suggested that perhaps we could block CGRP effects post-junctionally and have something that was perhaps better than triptans. And there was some work to support this. CGRP was measured in the draining jugular vein of the affected side. Quite often you have the pain in migraine associated with one side of the head, and levels reduce as migraine clears. And for example, if you treated with triptans, the CGRP levels would reduce. Also, it was shown that CGRP can induce a migraine in migraineurs. So that was an important finding as well. So, where did this fit in with what was known about the pain in migraine? Well, that's been debated for a long time as to how the pain occurs. People agree that the brainstem's involved, without a doubt, and also that there's three key factors. There's cranial blood vessels in the dura, which is a pain-sensitive area. There's a trigeminal sensory nerve containing the sensory nerves and their neuropeptides, and this interacts closely with those blood vessels. And then there's links with the brainstem. And there's obviously some GI side effects, etc., and that might be related to these links with the brainstem. So, the search was on for a CGRP antagonist. And the first one came from Boehringer, Alstegupant. And they called it a small molecule, a CGRP antagonist, but it was quite bulky, really. It was discovered through a high-throughput screen, and it was a dipeptide-like compound that they adapted until they got this prototype. You can see here it's quite bulky. It's certainly not orally active, but it was selective in humans. And they tested it, and this is an experiment they did in the marmoset. Didn't have a huge effect on blood pressure, but if you stimulated the trigeminal nerve and you got facial flushing, then this compound would inhibit it. And also, it was dose-related. So they did this in the marmoset, and it was quite pleasing for me at the time because they adapted their model to the marmoset from some work that I'd previously done with Helen Connor at Glaxo and with Jane Escott, who was one of my PhD students, where we developed this model in the rat for CGRP antagonists. Sadly, Glaxo shut their CGRP program, or CGRP antagonist program, before we could use their compounds. This is a bit of a tricky slide, I think. So they took patients who developed a migraine, and they got them to come to the clinic when they developed a migraine, and they looked at their response rate at 2 minutes later. And here we've got a number of patients on increasing doses, and it seems to work from what you can see here. What they did was add all these patients together and put them into one response rate and compared it to placebo. In migraine, the placebo rate is always quite high, but it seemed to be beneficial. It's really good news. People tried to make orally active CGRP antagonists, but this didn't seem to work too well for some time, and it was associated with some adverse effects in the liver. So we had this situation where we were pretty sure CGRP was released in migraine. We've got antagonists which combine CGRP and stop signal transduction. But was there anything else? And people were saying, what about anti-CGRP receptor antibodies, which perhaps bind to the receptor to stop signal transduction, or antibodies that just mop up CGRP and stop transduction? It was asked in very good faith because at the time, TNF-alpha antibodies were shown to most definitely work in arthritis. And indeed, some people here will remember the headlines. Expensive, but they worked. So why not try antibodies in migraine? The problem is, we thought, or I thought, they would have to cross the blood-brain barrier. So they'd have to, the dura's outside the blood-brain barrier, so that would be just about all right. But it's possible that to get into the tissues that were relevant, they would have to cross the blood-brain barrier. Antagonists could, but could antibodies? This was the big question. And the answer was yes. To my surprise, antibodies were very effective. So we've got 4 antibodies. They're all passed for use in 2018 onwards from a number of pharmaceutical companies. They're used today. Most mop up CGRP. There's one against the CGRP receptor, given by injections, and they have long durations of action. So this implies that CGRP modulates migraine at a site where the antibodies can reach. And the other thing that became immediately obvious, and perhaps you might expect this with antibodies, there are very few side effects. This is from Migraine Buddies, and this just shows these antibody treatments, but the CGRP antagonists are catching up. And there's several, more than I've got here, now available for oral administration. So I just see more and more positivity when I read reports from meetings because these CGRP blockers appear to be effective in various types of migraine and also as preventive treatments as well for those who have chronic migraine. There are some side effects, but they're quite a few. And overall, they're well tolerated. Constipation, but Peter Goadsby assures me this can be dealt with them without them coming off the CGRP blocker. There are some cardiovascular side effects which have been much debated, but not as many as someone like me would have expected, as it's such a potent vasodilator. But it suggests that endogenous CGRP may not be that important in the terms of the regulation of blood pressure, etc. There is one of them, the antibody to the CGRP receptor, that actually comes with a warning not to be used in patients suffering cardiovascular conditions such as hypertension. So what are the presumed mechanisms? That CGRP is released from the terminal, and that the CGRP antagonist, commonly called Japants, so I think pants is a bit sad, but that's the name they seem to have got, and under those conditions, we won't have any neurogenic vasodilatation. Alternatively, the CGRP antagonist will stop signaling in terms of neurosensitization and increased pain transmission. I get the impression more people think that this side of the equation is important, but whichever way you look at it, in migraine you certainly do get increased blood flow. So, where have we got to? We had a discovery of CGRP. It's a vasodilator. It's linked to migraine. Then we had a real problem, and it delayed us for years where people got the wrong receptors. Finally, we got the right receptors, and we realized that the CGRP antagonist works. Then we realized that the non-peptide oral antagonists had these liver adverse effects. That seemed to stop the field instantly. It was quite amazing to someone like me. Then it was realized the antibodies developed benefit of migraine. They were finally approved in 2018. Then there was a big push, which took a lot of energy to get them to be accepted by people like NICE. And now we've got to this wonderful situation where they're found to be effective in many types of migraine. So they're very much on the landscape now for treating migraine, whether they're the CGRP antagonists or the CGRP antibodies. There's lots of other treatments, but there's a lot of people who don't get effective benefit from treatments for migraine yet. So that's a success, I think, in drug discovery, and I think I'm very lucky to have been part of that, because of course you hear so many drug discovery programs that go wrong. But I just wanted to end with what we're working on at the moment. And I wondered, if you turn the concept around, will CGRP agonists be beneficial in cardiovascular disease? So we've already learnt that you can block them without any bad effects. But can you utilize them? The way we've approached it was to do studies with CGRP, but it has a very short half-life. So we worked with somebody, Net Sams, initially from Novo Nordisk, to stabilize CGRP, just like you stabilize insulin. And it's isolated with something that's associated with albumin, and the half-life is changed from a few minutes to several hours. And what we've done is tried to understand what happens when you give this analog in a mouse model. First of all, it was angiotensin II-induced hypertension. There's basal blood flow. And then we raised the blood pressure with angiotensin II in a mini pump. And this is what you would normally see if you carry on. But if you give the CGRP analog on a daily basis, you get a reduction in blood pressure. From this, we went on to study and see if it protected against heart failure. And first of all, Aisha Abdul did this work. We challenged mice with abdominal aortic constriction. That's an overload model. And looked to see how the mice benefited. And they seem to with the CGRP agonists. We've extended the studies now, and we've got 4 distinct murine models that show a therapeutic potential of the CGRP analog. They protect against the onset and hypertension and cardiovascular remodeling. They seem to be effective over a long time, and there's a lack of obvious side effects. Also, they seem to be beneficial on a long-term basis when we just give them for a few days. So, we've done an awful lot of work. I don't expect you to look at this, but I just wanted to point out just 3 things that we've done in terms of echocardiography. The ejection fraction of, if you look at the black, of the AAC mice goes down. These are the mice with the overload model, and it comes back with treatment. Septic wall thickness goes up in heart failure. It reverses with our compound. AAC is the long-acting CGRP agonist. And also the swelling, or it could be fibrosis in the heart, is reversed as well. So, to conclude, I'd like to say there's been a real rich history of sensory nerves and their biology. And in pharmacology, we've made a lot of use of it, I think. The neuropeptide research area has had its ups and downs, but I believe CGRP blockers have changed treatment paradigms in migraine. And certainly, I'd like to suggest that if we can have stable CGRP agonists that are orally active, that may be useful in diseases such as heart failure. Finally, I just wanted to, this is not an exhaustive list of all the people who've sort of worked with me to try and understand where sensory nerves fit in, but I'd like to thank the people over the years who've worked with me, especially the people at the top who've worked on the cardiovascular models. So, thank you very much.
So thank you very much, Sue, for a real tour de force around sensory neuropeptides and sensory nerves and their role in physiology and pathophysiology. And it's a really good example, I think, as you say, of basic science translating into drug discovery often, which doesn't happen for various reasons. So, are there any questions to Sue from the audience? So I think, Sue, just before we have got somebody, I'll ask you the first question, if you don't mind, is you touched on the end about using CGRP agonists, and you can show some protective effects in the cardiovascular system, but earlier you told us that some of the early evidence that CGRP is involved in migraine, that if you infuse CGRP into migraineurs, they get migraine. And I just wondered, at the moment, if it's preclinically in a mouse, they can't tell you about migraine, but do you think that is a real concern, particularly if you've got a long-acting agonist of CGRP?
Yes, it's always a worry. We have got light dark boxes, which are used by migraine biologists for studying migraine. And when we, I took that slide out, but when we use our analog within those boxes, the mice seem to act normally. So at the doses we use to get this cardiovascular protection, there's no evidence that the mice want to go and sit in the dark, which with some migraine stimuli they most definitely do. We're now into larger animals, and the suggestion is they look happy, but of course we won't know till we get to humans. I agree with you.
Yeah. Okay, so we have a question over here at number 4.
Thank you very much for your talk. An amazing body of work. I have 2 questions, hopefully neither too long to answer. First is actually going back to some of the work around that you talked about around TRPV1. I just wondered if you have any thoughts on quite a lot of the research around cannabidiol has produced negative results for pain management, and whether you think, given that that is a TRPV1 antagonist, whether that bodes poorly for further research with specific antagonists. And then second, any thoughts on the use of anti-CGRP treatments for other pain conditions, for example, osteoarthritis, which I think there's been some trial evidence which has been negative for as well, but interested to know your thoughts on why that might have been negative.
Right, yes. I gave a very simplified version of the TRPV1 receptor. So there's various different types of stimulation that definitely happen to that receptor that leads to pain, and the capsaicin-induced stimulation, which leads to the desensitization, is only one of those. So I don't think you can fully predict until you get to the point where you examine your compound in your clinical study or your preclinical study. And of course you can use sort of NK1 or TRPV1 knockout mice and things like that. So that's a really difficult question, because the TRPV1 activation syndrome is, yes, you can split it up into multiple mechanisms. With the CGRP antagonists, now they've got these drugs and they've got such good evidence from migraine in a lot of studies. The real world evidence is out there and it's good. People are most definitely looking in other syndromes, especially neuropathic pain, but also arthritis. There was one, Lilly did one study in osteoarthritis, and I think they did a very short study because they thought it would work, and it didn't work. And a lot of people have discussed that study since and asked whether they stratified the patients enough, et cetera, et cetera. So people haven't given up with arthritis. Now they realize these drugs are good. But if anything, they're also going into post-operative pain, et cetera, et cetera. Yes. So does that answer your question?
It does. Thank you very much.
Gilberto.
So great talk. People use clinically beta blockers to prevent migraines. Have you got any idea about the interaction of beta blockers and CGRP?
So people have tried, definitely tried combination therapies. My understanding, and I may be wrong because I'm not a migraine expert, is that people tend to put people on CGRP antagonists or antibodies when they haven't worked, when previous drugs such as beta blockers haven't worked. But I'm sure there has been a combination treatment somewhere, but I don't know what happened there. Yeah.
Okay, Sue, well, thank you once again for an excellent lecture, but we have this room for another lecture afterwards. But before you go, Sue, it's just my duty to present you with a certificate. So, and I guess they want to take a photo of you. So thank you very much again, Sue, for a really excellent Paton lecture this year. And yes, it's an amazing amount of work, and I wish you well with the rest of your career. And for those of you who are waiting for the oral session, I think it starts in about 10 minutes in this room. So thank you for your participation. Thank you.